Hi friends, today we discuss about the immunity and immunoresponse. Immunity. This immunity under the class of immunology, and this immunology which is the big subject, which is a specialized doctor is the immunologist. This is not discussed in one class. It is required at least 10 to 12 classes required for this discussion. But today we discuss the basic concept about the immunity and how to immunoresponse will be developed. And this is very important in this present situation because we are in the pandemic situation. The COVID-19, which is pandemically whole worldwide. What is pandemic? That means their spreadability in the world, whole world. It's called the pandemic. And basically this COVID virus, which is basically the less immune man affected by it. So what is immunity and how it will be developed? This is known to all everybody, not only this medical person but also the other students and others person also know how the immunity will be developed. Now we start the immunity and immunoresponse. The immunoresponse, the basic concept. What is immunity? It is free from burden. That means it is totally free from burden means, or it is a preventive action or the ability of an organism to recognize and defend itself against the pathogenic or antigens. That means to prevent, it is the ability to prevent the fight against the pathogenic microorganism or the antigens or any type of burden. It is called immunity. Now in the immunoresponse, it is the third line defense involve the production of antibodies and generation of specialized lymphocyte against the specific antigen. That is the immunoresponse. That means when the immunized person, there is some responsibility is called the immunoresponse. Next, antigen. What is antigen? The antigen molecule from the pathogen or the foreign organism that are provokes the specific immunoresponse. Basically, the antigen are the foreign substances, basically in the protein in nature, which is coming from the microorganism or in the foreign substances which stimulate the antibody or immunoresponse. Immunosystem. Their immune system is the third line of defense against infection. First one is the non-specific defense mechanism. There is no specificity of mechanisms and specific defense mechanism. Another is the immunosystem. This is the in our body there is some immunosystem. There is a specific defense mechanism, but in the non-specific defense mechanism is the first two type. Only the first line of defense, and there is a second line of defense. What is the first line of defense? Then our first type of defense, which is non-specifically determined by the skin, mucous membrane, and secretion of skin of mucous membrane. Basically the skin which is the mechanical barrier. That's why our skin, our body is covered by the skin. It is a mechanical barrier to prevent the attack of organisms like bacteria, virus, fungus or any other organisms can be prevented by the skin. That's why our body is covered with skin. If it is not covered, or suppose your sometimes wound or not covered portion, that will be infection very easily, because this entry point of the organ will be entry point to open this, to open the or entry point of the organism. If it's some wound or cut, that's why when you wound or cut, there is a chance of infection is high. And then the mucous membrane which is in acidic pH. That's why when the organism which goes to the mouth, that mucous membrane can destroy or kill the microorganism. So their secretion in their skin also, there is some secretive material which secrete and lubricating action to prevent the attack of organisms. That's why the first line defense by the skin and mucous membrane and their secretion. And second one is the phagocytic white blood cell. We are already everybody is know the WBC or in the phagocytic cell, which is the defense mechanism by the fight against the pathogenic microorganism, and this fighting is called the phagocytic effect. Another is the antimicrobial protein. Some antimicrobial protein which also defends our body, and some the inflammatory response. Sometimes some inflammation is will occur, that inflammation which reduce the your infectivity or immune response capability develop. In the specific defense mechanism is the immune system, and this is called the third line defense mechanism, and this is done by the lymphocyte and the antibody. Later we discuss what is lymphocyte, which is coming from the another group of the WBC or white blood cell corpuscle. Another is the antibody, which is generated by the influence of antigen. You can see in later. Now the innate or genetic immunity, that means this type of immunity will be born against the organism inborn, that is determined by the genetically. Some person are genetically highly immunized person, some are low, that is depend on the genetic variation, and that will be come from the maternal side. The genetically determined, that means, and may be due to the receptor or other molecule required for the infection. Lack of receptor or other molecule required to infection. In this case, some infection which also aggravate the immunity, that is called innate or genetic immunity. Innate human immunity to canine distemper, and immunity of mice to poliovirus. Generally in case of poliovirus, it is immunized person. Acquired immunity that are organism developed during the lifetime, that is called acquired. That means you can acquire, or it is called passive immunity, in your lifetime by the environment you can achieve it, that is called the acquired immunity. It is a non-genetically determined, there is a no genetic influence, but you can develop yourself, and may be acquired naturally or sometimes artificially. Naturally means naturally acquired means there is some infection will be goes, then you are immunized, that is called naturally, and sometimes we are vaccination against the some organism, that is called the artificial acquired immunity. Or development of the immunity to measles in response to the infection of vaccination. Development of immunity to measles. In case of measles, the response is the infection, sometimes infection, somebody are infected by the measles, the second time there is no chance of measles, generally in 98%, but sometimes it will be developed because they are not properly immunized, and sometimes this vaccine, the artificial antigen which is incorporated into the body, that is also immunized by the person, that is called the acquired immunity system or acquired. There is some immune system, human immune system, that helps the immunity. First organ or immune organ, that is the adenoids, which is present in the brain, tonsil, lymphatic nodes, right lymphatic ducts, thoracic duct, Peyer's patch of small intestine, appendix, which is also the immunized person, thoracic duct, thymus and spleen, and lymphatic vessel and bone marrow. These are the components which is help for the immunity system and develop the immunity. Lymphatic vessel, blood capillaries, tissue fluid, intestinal fluid, and lymphatic capillaries, and messages on the lymphocytes and macrophages, which are the some organelles which are influence the immunity of individual body. So we can classification of immunity. Basically the immunity is the two types, one is the adaptive and the innate or adaptive or acquired. This is again classified into two types, one is the natural, another is the artificial. And natural can be developed by the passive, by the maternal. Sometimes one person or a child can be immunized from his mother to umbilical cord, that is called passive natural adaptive immunity. Another is the active, when sometimes the infection of the child or a person, that is, and they are immunized, that is active natural adaptive or acquired immunity. Another is the artificial adaptive immunity. It will be classified again two types, one is the passive, another is the active. What is passive? Passive means when the direct antibody, suppose a rabies antibody which is directly introduced, or tetanus antibody which is directly introduced into the body, that is called the passive immunity. Which is active immunization by the vaccination. That's why UNICEF and the WHO guided as when the child is birth, at the time there is some vaccine schedule provided by the hospital and the other healthcare system also, and generally in the birth the BCG vaccine is provided. Then after there so many schedule will be given to the patient children, they are by the immunization due to by vaccination. This is called artificial adaptive immunity or artificial acquired immunity. Natural immunity. This natural immunity can depend on the different types of different factors like species. Say for example, in case of this natural immunity is differ species, one species to another species. Say for example, man is susceptible to plague, but fowls are not. Man always susceptible plague, fowl are not. Fowl is never susceptible or never infected by the plague. Same times the race and an individual race, the mice are not affected by the typhoid fever, but it is the serious disease of humans. The race is a very important factor for natural immunity. An individual, some peoples are more resistant in the cold and skin condition infected than others. They are always seen. Suppose today in the class 50 students are come. After 50 students, there are some person are infected the common cold, but others are not. Why? Because there is some individual immunity. There is some different individual immunity. Some person are highly immunized to common cold, they are resistant, or sometimes the cricketers, they are going to today in one country, then goes to the next day they go to the another country. So their immunity is different. But when a simple man can goes to one country to another country, then immediately they are infected or infected by some organisms. That is the main things. But in case of acquired immunity, it is also two types. One is the active and the passive. Natural by the infection and artificial by the maternal. Maternal, in case of maternal, basically the colostrum, it is contain the high amount of antibody. This goes to the mother to children or children. That child can be immunized. That's why in this case, in this context, you can see, remember it, the mother in case of pregnant, it will be more immunized. That's why the food vitamin also required for a pregnant lady. If she is immunized, their child also immunized. So in case of pregnancy, their foods, their nutrient, their minerals and vitamins is very important, and take care it. And then the natural by the vaccination and the artificial by the antibody transfer. Direct antibody will be transferred to the body. These are the immune system. First, the pluripotent stem cell. What is pluripotent stem cell? Which is coming from the bone marrow. This pluripotent stem cell produce two cells. One is the lymphoid stem cell and another the myeloid stem cell. Lymphoid stem cell produce the T lymphocyte, which is most take part of the immunity system, and another is the thymus, which is produce the NK, that means natural killer lymphocytes, and other is the beta lymphocyte, or they produce the plasma cell. And in case of myeloid stem cell, they produce erythrocyte, and another the macrophages or blood clotting. Not only the macrophages blood clotting, but also it is used for the some immunity system or develop the immunity system. Monocytes, the macrophages, and the granulocyte. These are the T lymphocyte, B lymphocyte, macrophages, natural killer cell, which are also the seriously take part the immunity system, develop the immunity system of our body. Just we can see in case of blood formation, we can see how the blood corpuscle will be formed. Generally the pluripotent stem cell, which is very important, the formation of myeloid stem cell and lymphoid stem cell. This myeloid stem cell, which is the CU colony forming unit E and colony forming they produce ultimately the red blood cell, and the ultimate and CUFMG, which is produce the platelet, and eosinophil, which is for the eosinophil, and basophil myeloids, which are the basophil. You see when the myeloid and microblast and T lymphocyte and B lymphocyte by the lymphoid stem cell. Basically, this T lymphocyte and B lymphocyte, which is seriously take part in the immunization of this person. This is T lymphocyte and B lymphocyte. This B lymphocyte produce the plasma cell and the monocyte produce the macrophages. This is discussed in the, we can elaborately discuss in case of in your physiology in the blood formation. Immunity can be immunoresponse, the immunity and the immunoresponse system. And in case of immunoresponse, generally two types of immunoresponse. One is the cell-mediated immunity, another is the antibody-mediated immunoresponse. The cell-mediated immunity, in this case, when the antigen, that means the infection or the antigen or the foreign substances which stimulate the antigen-presenting cell, they produce the virion cytosine cell 1 T cell and virgin helper T cell. These always they develop the effector cytotoxic CSL1 and memory cell and effector helper T cells. That means in this case there is a two common. One is the memory cell. This is very important. Memory cell, which type of cell? Just like in your memory. Suppose sometimes we are discussing in the class some question and you can memorize it. When I ask in the next time, you can answer me in your memory. That is very important. This memory can help the answer for the future. So in that case, same type of memory cell will be generated when some antigen which introduced into the body, there is some memory. If the memory cell is developed, that memory cell can fight against the invading antigens. So memory cell, if the increase the number of memory, suppose your memory will be increases, your answer to try to give more answer. So if the memory cell, they can recognize immediately the antigen, same antigen, and it can prevent it. So the memory cell is very important, and ultimately the target organ, the cell with the antigen, and on the surface interaction pathogens and outcome, the cell killed directly by the memory cell. Another is antibody mediated response. Antibody mediated, here not said to be antibody, here is the antigen same. When the antigen is stimulated, there a virgin B cell will be stimulated. Here it is the T cell, here is the B cell, and sensitized to B cells, they produce memory B cell and effector B cell, and ultimately this effector B cell produce direct antibody, and this antibody target the pathogens of the toxin and circulate to the tissue and extracellular pathogens, and outcome the direct inactivation and direct destruction of the pathogenic microorganism, basically the production of antibody by the B lymphocyte. In case of cell mediated, general T cell and the general different types of T cell, T helper cell, T natural killer cell, and other cell. So around 5 to 6 types of T helper cell will be produced by the cell mediated immunity. That's why it is called the cell mediated immunity. But in case of antibody, or it is called the humoral immunity, by the B cell they produce the plasma cell, and in case of T cell they produce the memory cell. So in this case, immunity and the immune response, in this case there is some pathogens, bacteria, fungi, virus and foreign body that can stimulate by the B cell. That B cell maturity plasma cell, this plasma cell ultimately secrete, and this antibody bind with the antigen, ultimately the virus can be killed. Another mechanism is to mature into the active killer cell or T cell. The cellular immunity will be T cell, cytotoxic T cell, and the virus infected cell, the kill the reaction. Or another is the helper T cell or lymphokinase, this lymphokinase or lymphocyte, which is also kill the microorganism by the, actually in this case I convey the message also. Actually the antigen is the very important, not among the antibody, because the antigen are the substances which are the stimulator. If the antigen is introduced into the body, then it can be stimulate the B cell, either T cell. That antigen is stimulate the B cell and T cell. Ultimately the B cell or T cell fight against this antigen. So antigen, that's why the antigen property, their chemistry is known very well. Otherwise you cannot say immunized or immunoglobulin or immuno type substance like vaccine cannot be produced. How because if the antigen is known, then you can design the antibody with the help of the structure of antigen. The cell mediated immunity, generally in case of macrophages, it is infected by this, the antigen MHC, major histocompatibility complex, major MHC, major histocompatibility complex. It called produce the memory T cell. They produce cytokines, or T helper T cell. They bind to the receptor. They produce cytokines. Another neurotransmitter, another chemical transmitter. They produce mitosis. Ultimately, the infected cell with the cytokines. These cytokines, generally in case of coronavirus also, this cytokines is very important to target the coronavirus and ultimately kill cell death. The cytokines, so the newer terms in the cytokines, chemokines, which there so many group of or interleukin which produce from the T cell, and they bind with the coronavirus and that affect the or kill the microorganism. Types of acquired immunity. Naturally acquired immunity obtained from the course of daily life, which is in your environment. Natural acquired immunity, and antigen or pathogen enter into the body by naturally, and body generated an immune response to antigen. In this case, body, when the antigen introduced into the body, that body can fight against the body, and the immunity may be lifelong. Sometimes you have say suffering from the chicken pox and mumps or temporary, but in case of influenza and intestinal infection, there is a permanent immunity will be developed, never you will be suffering from the influenza. Naturally acquired passive immunity. Antibodies passes from the mother to fetus and via placenta and breastfeeding colostrum. The first milk of the mother after the childbirth, it is called the colostrum. This colostrum which contains the high amount of antibody, and that antibody immunize the newborn baby by the breastfeeding. So that's why in case of first 6 months of child, babies can be profuse breastfeeding. This is the only diet for the new baby. It is necessary because this colostrum, this male breastfeeding is important for immunization and nutrient. All this given from her or from her mothers, and no immune response to antigen. Immunity is usually short life, week or month. It is not a long time, but it is basically the short period of time, and protection until children immune system can develop. That means when the immune organ which are not developed in born, which is developed generally first day up to 15 years of old. Before that, this mothers or maternal immunity can help the immunity person. That's why sometimes the children is suffering from the so many diseases, or they are affected frequently due to less immune, because their immune system cannot develop 15 years. Type of acquired immunity. Artificially acquired immunity obtained by the receiving a vaccine or the immune serum. And artificially acquired active immunity. Antigen are introduced into the vaccine. By the immunization, body generate the immune response to antigen. Immunity can be lifelong. Oral polio vaccine, if the oral polio vaccine is taken, it is lifelong immunized person, and temporary by the tetanus toxoid. When the tetanus generally 1 to 2 years, tetanus toxoid can be immunized. When after 2 years, when it is any infection by the tetanus, then always you take. That's why you have frequently take the tetanus toxoids. Among this, we are also know the what is antigen, antibody, toxoid. That will be, this definition is very important for your examination also, and other parts also. Artificially acquired passive immunity. The performance of the antiserum and introduced into the body by the infection, and the snake antivenom injection from the horses or in the rabbit. And immunity is short life, half-life 3 weeks, and short immune system does not respond to antigen. Serum. What is serum? Now the fluid-like remains after blood has clotted and the cell have been removed. That is called the serum. That means cell-free plasma is called the serum. but serum and plasma is quite different. It is plasma contains some plasma protein. When the plasma contain plasma protein and others, but not cell, but serum is totally free from totally corpuscles. Antiserum, this is also another term which are used in the pharmaceutical. The antiserum, the serum containing antibody to the specific antigen obtained from the injection, and the animals like horses, rabbits, goat, and with the antigen, snake venom, botulism, and diphtheria toxin also. Serology, this is the most important test. Serology, it is the study of reaction between the antigen antibody. That means the antigen antibody reaction study is called the serological study, or it is called commonly the serology. In case of dengue also, there is sometimes government order the serotype, that means the dengue positive. A gamma globulin, another is the fraction of serum that are contain the most of the antibody is called the gamma globulin. And serum sickness, which is the another disorder or disease caused by the multiple injection of anti-serum. Sometimes we are injected the multiple serum, this immunoresponse of foreign protein that may cause fever, kidney problems, and joint pain, and rare today. But this serum sickness is rare today. But if simultaneously more than one antiserum is incorporated into the body, then there is a chance of serum sickness. But nowadays, duality of the immune system. Duality of the immune system means, in case of human system, the humoral antibody or B cell, it involves the production of antibody against the foreign, and the antibody are produced by the absence of lymphocyte and the B cell. The B cell are immunized and the active present by the plasma cell and extracellular fluid, blind plasma lymph, and also the defense against the bacterial toxin, viruses that are circulate freely in the fluids and before they are enter into the cell. Also cause the certain reaction against the transplanted dead tissue. So antibody are produced by the B lymphocyte. How it will be formed? They generally formed from the bone marrow or the fetal life. This is coming from the pluripotent stem cell, which is already discussed in the earlier. And this lymphocyte stem cell, which is produced the thymus, and thymus produce T cell, or lymphocyte directly produce B cell. Ultimately T cell and B cell, they produce lymphoid tissue or lymph nodes, spleen, and blood, and they produce the antibody produced by the B lymphocyte, and they the produce of humoral or antibody mediated or humoral immunity. Duality of the immune system continued. The cell mediated immunity, whereas the first one is the humoral, then it's a cell mediated immunity. That means here is some cell which is coming from the T cell, T helper cell, T B cell, natural killer cell, or cytotoxic T cell. They are immunized by they produce the memory cell, and that memory cell can immunize the other person, immunize the body. And T cell regulate proliferation and activation of the other cell immune system, and B cell macrophages and neutrophil. And the defense against the bacteria, virus, and other inside host cell are insensible to the body. Fungi, protozoa, helminthes, cancer cell, and transplanted tissue also. Cell mediated immunity here, the same type of bone marrow fetal cell, they produce lymphocyte and thymus T cell. Ultimately the lymphoid bone marrow tissue, they produce the cell mediated immunity from the T cell. Basically the T cell here is the main function is the T cell, not the B cell. They produce the memory cell. So there is a difference between the cell mediated immunity and the humoral immunity. In case of B cell, generally humoral versus cell mediated. In case of B cell, is humoral, is the intact antigen, they produce the plasma cell or memory B cell, they fight against the organism. This antigen engulf and displayed the phagocytosis T cell. This antigen displayed the infected person, they produce the memory cell, and memory cell TH or TH means T helper cell, T cytotoxic T cell, and cytotoxic T cell active cell. The defense against the infection cell of cancer and the transplanted tissue also by the cell mediated cells. But in case of humoral cell secreted antibody that defends against the extracellular pathogen. Basically, in case of humoral system, the extracellular pathogen means bacteria, virus can be killed by the humoral system, but the cancer cell and the other transplant tissue which are by the cell mediated. So you can write in the cell mediated immunity or the humoral immunity or the beta cell that we can discuss in the earlier. So it is the chart which given to help for the student for humoral and cell mediated immunity, their characterization, their definition, their pathogens, main cell activation and others we discuss. You can see it, just you remember it, we discussed in the earlier, but here is the summarized portion of this humoral and cell mediated immunity. Antigen. So in this case, immunity can be developed or immune response can be developed, not the antibody. Most important is the antigen. If the antigen introduced, then that antigen it stimulate the beta lymphocyte or T lymphocyte, and that B lymphocyte and T lymphocyte they produce some mediator cytokines to another different cell. Ultimately, they produce the antibody. So the antigen is the key factor for immunization or immune response. This antigen generally in protein in nature, sometimes microbe in the capsule, cell wall, toxin, viral capsid and flagella, they are the toxin which produce antigen. And the microbes, the pollen, egg whites and the red blood cell and the surface molecules and serum protein and serous molecule from the transplanted tissue. And the lipids also, sometimes also the lipid and nucleic acid which is only not only antigen when combined with the protein and polysaccharide also. And molecular weight more than 10,000 and the higher. And the heptane, which is the small foreign molecule that will not antigen but must be coupled with the carrier molecule to the antigen, and once antibody are formed they will be recognized by the heptane. So heptane is a very important molecule, a small portion of molecule which is present in the inside the body. Epitopes, a small part of the antigen that are epitopes is the small part of antigen is a big molecule, proteinous molecule, but that are not bind with the cell receptor, but their small portion which is bind with the cell receptor. This portion is called the epitopes, and that's why antigen sometimes more than one epitopes. The small part of the antigen that interact with the antibody, it is called the epitopes, and any given antigen having the several epitopes, and each of the epitopes is recognized by the different antibody also. These are the side here, the A side and the B side and the C side. They are the antigen. There are three sides of epitope, antibody A antibody. That means in that case the antigen binding site, there is an antigen binding site. Here A can bind with the one antibody, B site can bind with another antibody, and C site which is can bind with another antibody. So there is a chance of whether their structure is also different. They are the epitopes and the antigen determining binds. So in that case the antibody binding site are the different. Here is the three different sites. If the same site there is a possibility of antibody can be one, but antigen is the different shape and size, different target area. That's why they are here to chance to fight three different antibody. Not only here three or more than three, it is also poly antigen binding. So antigen structure determine is very important for the production, for the preparation of vaccine. Antigen surface, antigen binding site is very important in their structure also. If you determine the structure in the recent study, in case of corona also, if the antigen is determined, then vaccine will be produced, their antibody will be produced. But unfortunately, this virus is totally undetermined. They are basically contain some protein or spike protein or non-NPS, non-structural protein, and that's non-structural protein which is non-structural, their structure will non-known, did not know. In that their structure will changes day by day. Generally at first the coronavirus coming from the three types A, B, C, but now in case of mutation they will produce the 11 types of coronavirus. So they are shape, their antigen, their antigen will change day by day. That's why their antigen cannot be identified. That's why the vaccine preparation is very tough and long time taken. If the antigen can determine, or protein structure can be, their virulence can be determined, then immediately or within a few months the antibody will be produced. But this virus, it so changes, it's just like the AIDS virus, their pattern is just like AIDS virus, and this spike protein, actually this spike, that means the spike protein which attack with the lungs, and immediately the lungs will be affected, and ultimately the lungs SRSA or severe respiratory acute syndrome will be produced. MRSA and SRSA, here the severe acute respiratory syndrome will be produced. That's why in case of corona, but nowadays the symptom will be changes due to their antigenic property will be changes. So in case of production of vaccine, their antigen determine is very important, otherwise you cannot determine the antibody. So antibody protein protects the recognition and binding of the particular protein antigen with a very high specificity of this particular antigen, and made by the response of the exposure of the antigen and one virus or microbes that are several antigen and determinants and huge different antibody, and each of the last antigen binding site, their variants of the antibody. So bivalent, trivalent or immunoglobulins, this belongs to group of protein is called the immunoglobulin. Antibody structure, the monoclonal monomer, or this nowadays the monomer flexibility, the Y-shaped molecule. So these are the antibody structure. It is generally Y-shaped in the structure. They are the SS bond, SS linkage. They are contain two chain. The one is the two heavy chain and two is the light chain. This two heavy chain which is bound by the SS linkage, and this SS bond which is bound with the antigen binding site, here is the purple color, and here is the antigen. So enlargement of the antigen binding site of the each of the epitopes. So there is an antibody or immunoglobulin. Generally 5 types of immunoglobulin is present in our body: IgG, IgA, IgM, IgD, IgN, and generally IgG, which is very essential in case of coronavirus. Initially the IgM is the high, then after few week it will be changes the IgG level. That means corona is positive, and this coronavirus will be identified by the RT-PCR or real-time PCR polymerase chain reaction. This is the positive test for this virus. These are the antibody immuno response after how it will be for when the vaccination or given. Generally first, when the vaccine will given, generally the first 7 days there is no antibody will be produced. After that, after 14 days the high level of antibody will be response. Then after 20 days it will be next goes down, then just goes to the baseline. Then after 20 days there is the second antigen exposure to the antigen A, same type of antigen, then it will be immediately goes to the second immunoresponse will be good, and it is just higher than the first, go fast, and then it's good. That's why in case of vaccination, first dose, second dose, third dose, and then that will be maintain the antibody level, and after that first, second, third, they goes to the booster dose. That booster dose can maintain the antibody level throughout the life, and at that condition there is no chance of being infected by the specific antigen or by the specific pathogen. So in this immune system, not only this, every vaccine will be this type of, but some little bit of variation. Here is the one vaccine blue color and that red color, but initially not grows, then second dose will be high, then third dose is high. Finally, if it's not going to the booster dose, there is a next after 3 to 4 years it will be fallen down. That's why the booster dose is very important to maintain the constant life of your immunity level as well as the antibody level, and that fight against the second exposure of second, third or fourth exposure of this particular organism. So today we are discussing about the immunoresponse. Next class we discuss about the immunoglobulin and how it will be defense against the defense against the or fight against the microorganism. Today to complete this topic. Thank you all the students. Or please, any question, please comment me in my comment box. If you like it, please like me, and if you understood very clearly, please subscribe my channel. Thank you all of us.
What you're given, that's how we live it. Don't be mad at the system, it's simply how we've existed. I hear a lot of people